lucky to have Trevor Pugh with us. The title of today's lecture is Pediatric Immunogenomics, Landscapes in Immunotherapy Prediction. And Dr. Trevor Pugh is a cancer genomics researcher and the director of the Joint Genomics Program of the University Health Network and Ontario Institute for Cancer Research. They deliver basic, translational, and clinically oriented genomic services. He's also appointed as the associate professor in the University of Toronto Department of Medical Biophysics, and he's a senior scientist at the Princess Margaret Cancer Center and a senior investigator at the Ohio Institute for Cancer Research. His lab is focused on understanding clinical implications of clonal shifts in cancer and non-cancerous cell populations during treatment. So, welcome, Trevor. We're excited to have you here today, and we look forward to hearing your lecture.
Thanks a lot, Jay, for the invitation and the introduction. Yeah, really thrilled to be a part of this webinar series. So today I'm going to talk about some work actually led by a postdoc in my lab, Arash Nabi, collaborating globally with Natty Jagger at DKFZ and Katie Hutchinson in Genentech. So there's going to be two sort of stories at play here. One, talking about what are pediatric cancer and what is an immunogenomic landscape, then trying to put this into practice and some forward-thinking ideas at the end of the talk based on what on the data. So some disclosures, the most relevant for this talk is this, we have some patents around a hybrid capture method for determining T and B cell repertoire. So yeah, today I'm going to talk about what is an immunogenomic landscape. It's a bit of a new idea. We then want to take that definition and sort of describe what do these landscapes look like and what do they what do they look like specifically in pediatric nervous system cancers. And if you want to look at many of the figures, we've released those in bioRxiv just tail end of September. So it's available there if you'd like to read in more detail. And then new work that is just currently under revision, an immunogenomic analysis of samples that were collected as part of the iMatrix atezolizumab study, and that's the work with Katie from Genentech. So yeah, I wanted to jump in and just talk about what is a landscape, and I want to start with this picture from Bob Ross, famous landscape artist, and really I guess some of the complexities of using the word landscape, because there's so many different ways to describe a mountain, describe foothills. There's interactions between water and the shore and soil and the beaches. So the spatial orientation and adjacency of samples is important. And all this complexity of looking at landscape paintings holds just as much for immunogenomics. So I flipped all the labels. So when we think about immunogenomic landscapes, we have to think of all the various pieces and aspects that go into an immunogenomic profile. So altered functional pathway and signaling, this could be somatic alterations. It could also be immune infiltration. Some of these interactions between immune cells. How do we measure immune response? Lots of ways to do that. I'm going to talk about mostly genomic technologies. And then we can't forget the somatic cancer genome that really kicks off the development of sometimes an abnormal landscape. So, of course, cancer landscapes don't look like paintings. They look like tumors. So this is the exact same figure, but I've swapped out the Bob Ross art for actually, it's really nice art actually by Ben Wang, who leads the immune profiling group here at Princess Margaret. And now, rather than looking at trees and mountains, we're interested in chromosomes. We're interested in how pathways are activated, which pathways are being used by both cancer and immune cells within a tumor. Measurements of those microenvironmental interactions, and I'll have several slides specifically on how do we measure immune response, and that's going to be using that T and B cell repertoire sequencing technology. So these specific technologies we're going to talk about, whole genome sequencing, and this is really driven by multiple and really excellent data sharing platforms and actually commitment from the pediatric cancer genomics community to make this data widely available. Same with whole transcriptome sequencing. So tissue-based imaging work. So this is immunohistochemistry with Paul Sorensen and H&E image analysis with Derek Oldrich at CHOP in Paulson, BC. And then some of the TCR and BCR receptor sequencing, and even reusing some of the existing genomic libraries that were already made for another purpose. The future is almost unquestionably measuring all of this using spatial single-cell multi-omics technologies. And actually, it's great to hear that Claudia is going to be giving a talk on that, because all the work I'm going to talk about is using bulk genomic analysis and trying to infer all of this information from existing sequencing data from hundreds of pediatric nervous system tumors. And why do we want to do this? It's really this thinking, and certainly we've done a lot of work in the adult arena around using knowledge of the various immune components that make up tumors to guide selection and potentially development of different immunotherapeutic strategies. And this review actually does a great job of conceptually orienting different classes of immunotherapy. They use the term passive and active. Passive is essentially providing missing parts. So if a tumor has evaded the immune system or has sculpted the immune system in such a way that it doesn't have any immune infiltration, you may want to consider different immunotherapeutic strategies compared to certainly the most famous immune checkpoint blockade, where you're reactivating existing components that are already in place. So that's sort of the framework overall, thinking about what makes up the immunogenomic landscape, but ultimately, what can we do about it once we've seen that painting? So some more Bob Ross art. This is now multiple different pictures, multiple different landscapes. And the question we want to answer going in, what landscapes are encountered specifically in pediatric nervous system tumors, which is an interest in the lab. So what is the paint? In this case, the paint is data sharing, RNA sequencing data from over 900 treatment-naive pediatric tumors across 12 different cancer types. We wanted to come up with a framework that could cut across different cancer types and really being driven by 3 different public data sets. When we started the project, it was called CBTTC. Now it's the Children's Brain Tumor Network. And I've listed all their websites here. Great collaboration with Natalie Jagger through the PedBrain Tumor Project, and they're part of ICGC, also with Stefan Pfister. And then a project I know very well from my postdoc days, we went back to the well, looking at the neuroblastoma data from NCI Target. And here's the rough breakdown of the different cancer types across different cohorts, and I'll have different representations of different cancer types. You'll see some of these little codes throughout the project, and I'll try to call them out as we go into specific cancer types. Meningioma, schwannoma, ATRTs, ependymomas, MBLs, neuroblastoma, medulloblastoma. But we'll encounter some of these little codes as we go through, and they're also defined in the bioRxiv paper as well. Not to harp on the importance of data sharing, but this really could not have been done without the Cavatica platform. So they've done a really great job of housing and federating all of this data across multiple projects all into one place. So we're able to essentially speak the same genomic language and have access to substantial compute to process these large number of samples. And I actually just pulled these numbers today from Gabrielle Miller, Kids First. Just hugely impressive storage footprint and data footprint that they've just amassed over there. I also want to acknowledge at this point Dragon Master, who paid a large fraction of our cloud computing bills as well. So that was a very important piece of support from them. We wanted to return in kind, so I put some links down here at the bottom where we wanted all of our work to be fully accessible and reproducible. So some links to the primary data in this specific workspace we defined on the Cavatica platform, access to the scientific computing code to reproduce all the figures that you'll see both in the papers and in this talk. And a new experiment for the lab, which is using CodeOcean, which essentially gives you a web accessible piece of code to essentially reproduce all the work at a single click within a web browser. So you have access to the data and the code all in one place. So yeah, I encourage people to check out those as well. So into the science. Data scientists' major #1 job is data cleaning. So our first step was to make sure that we had, especially as we were bringing in cohorts across multiple studies, we wanted to make sure that they were representing the same type of tumors. And this is just one exercise we went through with the CBTN tumors, specifically removing cell lines. We wanted to be focused specifically on primary tumors. In some cases, there were multiple tumors from the same patient. We only wanted one. We then only wanted primary tumors. We didn't want any of these serial tumors specifically for the study. These are extremely interesting. We might actually be looking at these as part of a follow-up study. We then wanted to focus on I mean, all pediatric cancers are rare, but we wanted to have tumors that are some of the more common pediatric tumors. So we want to have a sufficient sample size, then cross-reference these with pathology annotations that came along with each sample. And we ended up with 581 for CBTN cohort at the end of the day. And this is really why we wanted to share this data back on the Cavatica platform, just so no one has to go through this sample selection exercise again. And I just want to have a visual depiction of the effect of this. So we took all the RNA sequencing data and did unsupervised gene clustering. And down at the bottom here in colored blocks are the pathology annotations. And you can see there's little gaps, there's little tumors that are not necessarily clustering along with the rest of their cohort. And the effect of that filtration strategy was really to give us a nicely cleaned up. I don't want to say it's a homogeneous cohort because there's a lot of texture within each of these cancer types, but at least we're doing an apples to apples comparison, and the pathology interpretation is totally in line with the transcriptional cancer type inference as well. So this is an important upfront first step just to get all these data to play well together. So the goal here is immunogenomic landscapes. So we want to first define the parameters of immune cell infiltration. Which tumors have immune cells infiltrating them, which ones don't. A lot of these are brain tumors. We're not expecting extraordinarily high infiltrate levels. And we're using tools that were not necessarily made or tested in pediatric tumors. So we wanted to use this estimate immune score algorithm. This is widely used in the bioinformatics community. And we did an in silico dilution series. So in this case, it was a medulloblastoma patient-derived xenograft. And we computationally mixed these with a healthy spleen from the genotype tissue expression project. So we essentially did that dilution. So the percentage of immune reads or the percentages of reads from spleen. And what we wanted to do is generate this curve on the right that calibrated an estimated immune score to some estimate of the absolute number of immune reads present in each tumor. So that was an important upfront bioinformatics exercise, but then we were able to apply this curve across the entire cohort. So the y-axis here is percentage of reads mapped to immune cell signal. Here are all the cancer types here along the bottom. And then we just put some adult solid tumors, GBM probably the most relevant given the strong brain component in our cohort. But we just gave that cohort essentially as a secondary comparator group. And what we found were the highest infiltrate levels were in the extracranial entities, perhaps not surprisingly. We know the brain is immune excluded largely, but we're also able to recapitulate the work of other groups. So specifically ATRT having the widest level of variability. And that was what really struck me when Rash first showed me this plot, was it wasn't like every tumor had a very consistent, tight immune distribution. These were extremely wide, both across cancer types, but also within within cancer types as well. So ATRT actually just reported just over a year, maybe 2 years ago, that there are molecular and immune infiltration subgroups, and we believe we're seeing that in the data, and we'll go into that cancer type a little bit later. Same story with neuroblastoma and other tumors, just these high levels of variability, and really the benefit of unbiased immunogenomic profiling to get you a very quantitative measurement using RNA sequencing data, and also the ability to compare across other cohorts like we did with the adults. The other theme, and we'll return to this several times in the talk, is how important orthogonal validation is. And this is certainly an important part of putting together a manuscript, of doing discovery in RNA sequencing data, but then validating it using other samples and other technologies. In this case, we teamed up with Paul and Alberto at the BC Cancer Agency. They had access to a tissue microarray from Children's Oncology Group, which they stained for T cell and B cell markers. And we really found the exact same thing when you do quantitative analysis of these images, essentially highly variable levels of infiltration in ATRTs. And same story here in the neuroblastomas. These are like highly variable levels of immune infiltration, even pre-treatment. And if you sum all these H-scores up across all the markers and compare them to the RNA sequencing, we see very similar patterns to what we saw using the RNA sequencing data as well. and really showing RNA sequencing is very good use of the tissue because you can recapitulate similar signal that you would get potentially from multiple sections of often limited tissues as well. So what I just showed was overall landscape. So that's standing at 2 or 3 feet away from a picture. We now want to zoom in and get into what exactly are these cells that are inhabiting these tumors. And we got a hint of it from that immunohistochemistry experiment where there were differences in T cell and B cell infiltration. So we then turned to some of our other favorite immune inference tools. And we wanted to do that same calibration experiment like we did with the bulk, with the bulk immune estimate from the estimate tool. We now wanted to get much more granular down into individual cells. What kind of surprised us was these tools really didn't perform the same. They had different models, they had different reference sets associated with each gene, and doing that dilution series instead with bulk immune reads, we now sprinkled in immune reads specifically from purified B cells, natural killer cells, T cells, etc. And you can see just from these curves, they're not necessarily performing in the same way. And rarely do we get a very tight correlation between the ground truth, the reads we put in, and the inferred method. And this has actually been well explored by the authors of the timer and timer 2 algorithms. And surprising to us, most tools actually infer the presence of immune cell signal in patient-derived xenograft samples. There is a risk here that we may be measuring some mouse content, but we're not getting that same signal across multiple different tools. And there may be the risk that the cancer itself is expressing some of the marker genes associated with each cell type used by these algorithms. And it's that last point that we really wanted to address. Is there a risk that the cancer cells themselves are confounding the immune cell signal? So we set out to build our own set of genes that were still specific to immune cells, but they lack the expression in cancer cells. We wanted this to be truly immune specific, but to avoid the confounder specifically by pediatric cancer cells. So this is a bit of a Herculean bioinformatics exercise, mining the protein atlas, available single cell RNA sequencing data, the estimates already built, or the list of genes already built into existing algorithms. This got us a list of thousands of genes. We then restricted these to only the genes that are being expressed in pediatric nervous system cancers, another round of filtering specifically for that cancer effect. And then we wanted to take those genes that are only expressed in immune cells, not in cancer cells, and assign them to specific cell types. And this was that last step, looking at, rather than bulk immune expression, the cell-specific lists, but now filtering out all the genes that may be confounded by expression by pediatric cancer specifically. And at the end of the day, we arrived at these 210 genes that were specific to 6 cell types that we were confident were going to accurately measure immune infiltration and the subtypes within the infiltrate and not be confounded, especially as we move across all the different pediatric cancers in our cohort. And so what did we find? We ran these same tools. We now have our own new list of genes, list of cell types, essentially using this list of high quality genes. And what we found were these 4 broad immune clusters: pediatric inflamed, myeloma or myeloid predominant, immune neutral, and immune excluded. And here's the breakdown of different cancer types down here in the heat map at the bottom. So in general, there were specific cancer types, meningioma, schwannoma, et cetera, that were more either pediatric inflamed or myeloid predominant. And down the left, medulloblastoma, ependymoma, famously not overly well immune infiltrated and scoring low, or scoring high, rather, for the immune neutral, immune excluded cell type, or immune subtype. So return to that need to validate. So we turned to Derek Aldrich at CHOP, and we wanted to validate these immune clusters. In this case, he had just set up a really great framework to do quantitative image analysis of H&E slides. So he had 355 of these. These were all being shared through CBTN, and was able to give us a very quantitative tumor infiltrating lymphocyte score. So he had pictures like these with high levels of infiltrate, moderate levels, and low, and essentially recapitulated what we've seen from the RNA immune inference story. Essentially, the 2 groups that are associated with high inflammation, You also see exactly the same signal when you don't grind up the cells, when you have them on the slide. And these differences were statistically significant. You can also see by the color here, these are all different cancer types being populated across each of the immune subtypes as well. So those didn't turn out to necessarily be a confounder. And that was the goal of the whole study, was to come up with an immunogenomic framework that could cut across cancer types. That being said, we did want to look at the molecular subtypes within specific cancer types. So I mentioned this ATRT finding from a few years ago, Marcel Kuhl and others. We essentially were able to recapitulate that finding. So specifically this highly immune infiltrated MYC and TYR subtypes. We stratified neuroblastomas into not amplified and amplified, very different representations of the immune subgroups there. So the high-risk MYC amplified cases not having high levels of immune infiltration, and so on and so forth. So it was really an opportunity to take the somatic genetics and put it alongside the immune subtypes. That being said, it wasn't a lock and key. It wasn't like there was a subtype and you were guaranteed to have runaway T-cell infiltration or B-cell infiltration. So the association is imperfect. So while there's sort of a trend towards specific patterns of immune infiltration given your somatic cancer genome, it's not a rule. It's a little messier than that, and it's really why it's quite important to profile both the cancer genome but also the immune infiltrate together and side by side to get a complete picture of pediatric tumors. We did look at some other potential sources of differences in different distributions across the immune subtypes. And really, the only thing we found was that these immune excluded tumors were enriched in tumors from males. And we don't have any causation here. We don't know whether this is because there's specific immune cell subtypes associated with being male and therefore developing immune excluded tumors. We did also check ethnic background. We really didn't see any differences in distribution across the immune subtypes there. And we didn't see a difference across at age of diagnosis. So really returning to that idea that there's not one necessarily external feature that dictates where a tumor is going to develop or fall within these 4 immune subgroups, apart from being male, which may have some additional confounders, especially cancer type or cancer location. Another famous biomarker, certainly in the immunotherapy world, is tumor mutation burden. In our hands, tumor mutation burden as a continuous variable wasn't associated independently with each immune subgroups. So just showing here, essentially looking only at single nucleotide variants, including the indels, no big difference across the 4 groups. Where there is a difference is when you look way up at the extreme. So these really, really high mutators, pediatric cancer is famously low mutation rate cancer. But if you look at these really high mutators, you do see that enrichment specifically for the tumors that do have immune enrichment. So tumor mutation burden is very helpful as an extreme biomarker when you're above a specific threshold, but these subtleties, especially in these low mutation rate tumors, TMB really wasn't necessarily sculpting or associated with any of the 4 immune subgroups. And similar story for driver mutations. We looked at all the driver mutations, and over here on the right, we've actually classified them into various pathways. And essentially what we saw were these driver mutations cut across cancer types. They cut across the 4 immune subtypes. So we really didn't see a relationship between what was actually driving the tumor and the ultimately responsive immune microenvironment. And this perhaps isn't necessarily that surprising. Driver mutations do not that frequently actually encode, antigenic peptides, and in some cases, actually, it may not even be mutant peptides at all. They're actually driving an immune response. There may be more likely that some of the signals coming from out of context expression of non-mutant sequences, such as cancer testis antigens or other transcripts that may be abnormally expressed. So we weren't able to really draw a direct line between the presence of mutation and the development of any of these 4 immune subtypes. And that's kind of why this heat map is essentially a random checkerboard pattern. We did want to understand what was driving these gene sets. So we took the gene list associated with each of these 4 types, and we essentially asked what pathways are these genes encoding, and really turned up several pathways that have already been associated with positive immunotherapy outcomes. So these are all pre-treatment tumors. They're not treated, so we actually don't have any knowledge of immunotherapy outcomes for any of these patients. But we are seeing pathways like interferon gamma, which is one of the most robust signatures that held up very well as a predictor of positive immunotherapy outcomes, which speaks promisingly specifically for the pediatric inflamed cohort, potentially thinking about using some of these signatures to select patients specifically for immune checkpoint blockade. And I'll return to that story when we get to the second part of the talk. Going back to the need to validate all this, there was some really nice work using mass spectrometry analysis of the same tumors that are also in were also in our cohort, and we essentially saw the exact same thing. The same signal that we saw using the RNA subsequently validated, and so essentially you see a rightward shift of essentially the proteins associated with all of these pathways, specifically in the pediatric inflamed and the myeloid predominant subtypes relative to the 2 colder cancer, the 2 colder immune microenvironment subtypes. We now want to drive even further. So we looked at total infiltration, we looked at cell types, we looked at pathways. Now we wanted to look at individual genes. Is all this being driven by 1 or 2 genes? The answer there is essentially no. So we did gene set specific analysis on each of the 4 subtypes. Perhaps not surprisingly, pediatric inflamed, runaway upregulation of cells associated with B cells and granulocytes. Myeloid predominant, actually only 1 hit in this population being overexpressed, specifically macrophages, so associated with the myeloid lineage, and the flip. When you get to immune neutral, downregulation of J chain B cell associated genes, and immune excluded downregulation of granzymes J chains. So really the story is being supported in multiple different ways by looking at this data through multiple different lenses. And here's the heat map of various genes along the bottom, really being driven largely by what immune cells are infiltrating these tumors. So most clinically relevant, what about survival? So these weren't quite as clear in this case. This is confounded a little bit by the fact that these tumors, despite potentially sharing common immune profiles, are all being treated and staged in totally different ways. So this is going to need probably a cohort by cohort analysis or potentially trials testing each of these individual survival patterns. But we were able to conclude the infiltration of specific myeloid patterns provide a greater survival advantage than generalized hyperinflation of multiple cell types or low infiltration overall. So really what you want is a very targeted anti-tumor response. And there's a bit of a question of whether these signatures are going to be prognostic or could they be used in a predictive way if you're able to intervene specifically with a cancer immunotherapy as well. And that's the type of use we'd like to encourage from this data set. Could you predict or use some of this thinking around patient selection for a potential future clinical trial? We wanted to go beyond individual genes and specifically try to understand not just T cell function, but also the T cell repertoire. So this is where my declaration of conflict of interest came in. We invented this hybrid capture-based method that essentially, rather than using PCR to amplify T cell receptor loci, uses hybrid capture baits against the essentially the portion of the J region right next to a rearrangement as these T cells mature, depletion of the surrounding non-rearranged signal part of the genome, and then V capture. And what we want is this little molecular barcode that includes the B, the J, the V, the D, and the J, which essentially confirm the antigen specificity of these T cells. And at the end of the day, that gives us these little sequences with a VDJ combination, and we can count those to infer the diversity of T cells infiltrating a tumor, but also the presence of specific clones that may be expanding and potentially be anti-tumor. The other reason we liked hybrid capture is we're able to apply hybrid capture to existing DNA and RNA sequencing libraries. So we don't have to go back and get more tissue. We can just take libraries that already exist, potentially generated for another reason, lift out the TCR sequences, and do a clonality and a diversity analysis. We actually just had a paper come out yesterday in Life Sciences Alliance, the exact same idea, but applied to the immunoglobulin or the B cell receptor loci as well. So another technology that the dream is we'll combine all these base sets together and get a complete T and B repertoire in a single experiment. But for the purposes of this talk, I'm just going to talk about the TCR repertoire. So we had all this bulk RNA sequencing data, and we wanted to compare that to our CAP-seq data. And essentially, we saw a relatively good correlation when the diversity was very high. So if you have a lot of low-frequency polyclonal T cells, and we see them in our deep cap TCR sequencing data, that's essentially recapitulated in the RNA sequencing data. Where the RNA sequencing data falls down a little bit is when you have a clonal expansion, because the RNA sequencing it's relatively shallow at the T cell receptor locus. So you're only sampling at the very, very top of the repertoire. So you don't get this full diversity metric. But we were confident that we were able to separate clonal from polyclonal infiltrates. So whether our challenge is whether we could really pinpoint precise measurements of diversity, but we were already able to say this tumor has a more clonal infiltrate than some of the others. And here's the output of applying the TCR diversity inference to the RNA sequencing data, and really reflecting some of what we already knew from the immune infiltration levels. So neuroblastomas being highly variable, but also having a mixture of very clonal infiltrates and very polyclonal or oligoclonal infiltrates as well. And this is what some of the real data looks like. So if you look at the Shannon diversity and map it to the number of reads, one concern we had was, is it just the number of reads that are driving clonality? And there is some relationship there, but off of the diagonal, there are a large number, in this case, we've called it orthoneuroblastomas, that even if you have a large number of reads mapping to TCR, the diversity is not there. So these are the clonal populations. And contrast that to some of these tumors, also with middling levels of mapped RNA sequencing reads, but almost every read mapping to a different TCR sequence. So that really gave us the ability to distinguish these polyclonal from clonal infiltrates. And probably like you'd expect, the pediatric inflamed basically have a large amount of immune infiltrate, and it's not clonal. It's a ton of many, many different T cells that are inflaming these tumors in a generalized way. Myeloid predominant, these are slightly less diverse, and not surprisingly, very hard to find TCRs inhabiting the immune excluded and the immune, what we call the immune neutral. If you just look at the compare neuroblastoma to the CNS tumors, there's a marked difference in the extracranial versus the cranial. So really the context actually matters quite a bit here, with almost an order of magnitude fewer TCRs found in the CNS versus in the extracranial tumors, due to the CNS being immune protected. And these are some of the conclusions from this first paper, the one out on bioRxiv. So we have 4 immune microenvironments broken down across these different percentages. TMB on its own in the low mutation rate tumors was not independently associated with the inflamed microenvironment. We hypothesized that pediatric inflamed tumors may be good candidates for ICB, and the myeloid predominant may actually benefit specifically from this disruption of myeloid and lymphoid interactions, because the microenvironments of these myeloid predominant tumors were a mixture, not just of T cells, but also other myeloid lineage as well. And we did sort of note there is an anti-CD47 therapy actually being trialed, listed on clinicaltrials.gov now. So it'd be very interesting to do a biomarker analysis of that cohort to see if this hypothesis holds up. On the flip side, the immune neutral and immune excluded actually may not be the best candidates for immune checkpoint. The parts to the mainness not be in place to reactivate. And one strategy could be to accelerate movement of these patients into biologic immunotherapies to put the parts back in, adoptive cell transfer or CAR T. The other take home from that study was really there's few clinical surrogates for the immune subgroups. There's that very slight enrichment of males with immune excluded tumors, But what we really learned from the study was how important it was, just like we've been doing for precision medicine for cancer mutations, it's equally important to have a personalized immunogenomic profiling and really have these two pieces of data side by side as part of a complete cancer or tumor genome profile. So that laid the groundwork for our thinking of immunogenomics in pediatric and actually in adult tumors as well. We now want to take those landscapes and know which of the features within those landscapes, which of those pictures had the mountain that would tell us whether kids are likely to respond to immune checkpoint blockade. And this is where the collaboration with Genentech and Katie Hutchinson came in, really building on this fairly exciting, actually, phase 1/2 clinical study of atezolizumab for kids. And just like our original cohort, a mixture of tumors, solid tumors, and Hodgkin's and non-Hodgkin's lymphomas. Put the consort diagram just here on the left, so a fairly large study. Responses were not overwhelming, but it was an opportunity for us to try to understand why some of these patients were responding. And they actually put right in the abstract, they wanted this data set to be used and inform potential new patient selection methods. So it was really conceptually and a very exciting opportunity to look at some of these tumors from patients on this trial. So in the iMatrix study, they'd done PD-L1 tumor expression. They'd actually already identified that as a biomarker of response. They also flagged that it was a challenge having this mixture of lymphomas and solid tumors. And that's something we encountered when looking at the matched RNA sequencing data for these tumors as well. The other benefit in this study has been the existence of some immunohistochemistry data that we're also able to analyze alongside some of the other biomarkers that have already been developed. So yeah, we took a look at that immunohistochemistry data, very similar to as was done in the first landscape paper. And we found there was real value in scoring out specifically these T cell markers. So essentially the patients with high CD8 protein expression, both across the cohort, but also excluding the tumors that were taken from lymph nodes, lymphomas. We're still seeing this strong survival signal. The challenge with this study, just to draw your eye to the bottom, these are small numbers. So we're really thinking of this as a pilot study to essentially point us toward leads and help us validate some of our work in the landscape, in the landscape paper as well. Just as we did in the first paper, we applied the immune inference methods, applied them to the RNA sequencing data, and essentially recapitulated very similar survival curves, both in using across the entire cohort, but also when excluding lymph nodes. And likewise, the PD-L1 was already well established in that study, strong survival association with that specific marker, but also the ability to read it out from the RNA sequencing data as well. So really the opportunity of even bulk RNA sequencing as a major opportunity to score out and assess multiple biomarkers from a single data set. TCR sequencing, just as was done in the first paper, we were able to generate this TCR landscape using CAP-TCR-seq, in this case, applied to DNA from the baseline tissue samples. You can see huge diversity over here in the lymphomas. Not surprisingly, they're coming from these lymph nodes. And then this diversity of different immune infiltrate levels. So these ones down here on the left, low levels of diversity, so clonal infiltrates. And this one on the end, almost looking like a lymph node, just highly, highly polyclonal infiltrate invading this tumor. And we wanted to ask the question, is TCR diversity a useful biomarker in this context? And actually, indeed, it was. So TCR diversity at baseline, independent of atezolizumab response across the entire cohort, but even in the cohort once you take out the lymph nodes. But also, yeah, just be cautioned, not many responders in this study, but we were able to identify the patients with low diversity really not benefiting very much at all from atezolizumab treatment. So really a brand new biomarker because large numbers of samples had been banked and were available for molecular analysis as part of the trial. And very similar story. So every time we look, under a rock, we sort of uncover a very similar immunogenomic story. In this case, a differential gene expression between responders and patients with progressive disease. Again, that interferon gamma response pathway popping up over and over. So in this case, not just a prognostic marker, but potentially predictive in atezolizumab as well. And differential gene expression really driven very strongly by genes associated with immune infiltrates. So the take home from this follow-up study, immunogenomic analysis linked a lot of these potential biomarkers to atezolizumab response across pediatric cancers. This is a reality of a lot of these pediatric cancer trials. They're not necessarily going to be homogeneous populations, and really the need for analytic frameworks that cut across different cancer types. What we really learned was the multimodal approach is very powerful. It's hard just to look at immune cell or an immune pathway or just a driver mutation. And really, it's the combination of immune cell infiltrates measured potentially multiple ways, protein and RNA sequencing, and looking at novel biomarkers such as T cell receptor diversity as well. And similar to the first paper, how powerful the pull is for personalized immunogenomic profiling. And what we really need to do to bring these two studies together is to take the 4 subgroups that we found in the landscape paper and start to test those out in clinical trials, including this cohort here, and ideally larger cohorts as well, as samples are banked and profiled. So I just wanted to touch on a couple of future directions before I wrapped up. Really where we're going locally here in Toronto is supporting some pan-Canada precision medicine programs. The BC Cancer Agency has set up this, this protocol called Captivate, where they essentially score out 4 different aspects of the immune microenvironment, similar to as we evolved in our immunogenomic study. And here at Princess Margaret, Lillian Sue is leading a Marathon of Hope Cancer Center Network, network. And essentially the idea is to have not just genomic profiling, but also immunogenomic profiling as part of assignment of patients to both Captivate, but potentially some other clinical trials that are gene expression or immune cell inference driven. And to support that, the genomics program I lead at the Ontario Institute for Cancer Research has undergone CAP and Accreditation Canada accreditation, specifically for clinical reporting of whole genome and RNA sequencing. And we set this up, I just showed a little example of our clinical report on the left. We set this up originally for reporting mutations, fusions, and copy number alterations, but really where our activity now is moving towards this validating genomic landscapes for clinical reporting. And that's not just tumor mutation burden, which we already report, but other more subtle signatures that do sculpt the microenvironment, like microsatellite instability, homologous repair deficiency, immune deconvolution, some of the cell-specific immune inference methods that I mentioned throughout this talk. And really, this is really where I see clinical molecular testing has to go. It has to be comprehensive, and it has to be multi-omic, in this case, genome and RNA sequencing, because really only together that you get the full view of these immunogenomic paintings. So yeah, thanks everyone for coming. Full credit to Arash Nabi, who's really driven the relationships, the bioinformatics, the sample movement at the heart of both of these studies, and really call out the co-PIs, Nellie Jagger on the first study, great collaboration with the DKFZ team, collaborations with Derek, Paul, and Alberto, and the other co-PI for the iMatrix paper, Katie Hutchinson and the Genentech team. And a rich and longstanding collaboration with David Malkin and the Precision Oncology for Young People program funded by Terry Fox at the Princess across Canada. So thanks very much, and I'm happy to take questions at this point.
Thank you very much. I have some questions of my own, but I'm going to start from a few that have come in. This is a multi-parter, but the first one is, were there correlations between immune-filtrated tumors and immunotherapy outcomes?
Yeah, so that's the issue with the sample with the first paper. It was focused on primary tumors. Some of them were collected way before immunotherapies were even available, so we don't have immunotherapy outcomes. Our only shot at that currently is that iMatrix study, where we had a distinct clinical trial where we were able to make those inferences. I believe there's probably the opportunity to link all that banked data. This might be a question for Kabatica or the CPTN group. Is there a way to link that information and update the clinical data in case some of those kids did go on to cancer immunotherapy trials? So I suspect some of that data may be out there in some database somewhere. It's not sort of in the default annotation table that we got, but I think there's an opportunity there for a refresh of the clinical data in case some of those patients did go on to a trial.
Next question, have you looked at non-responsive hot tumor immune signatures?
Yeah, so these are very interesting, and you might have seen them on some of the plots. There are these huge outliers that don't necessarily have diverse T cells, or they have a very high immune content, but they're different from the other tumors with high immune content. So we don't have response data, but I think that is a very interesting subclass, essentially the tumors that break the mold of this pan-cancer analysis. So we haven't looked at response, but to me, that's the whole point, to be doing comprehensive profiling to identify those really biologically unusual tumors and to see if potentially they together might be an unusual subset that you could go after therapeutically. But yeah, we didn't have response in that first cohort, so we couldn't make that correlation yet.
Do you see T cell exhaustive signatures in the hot tumor population?
We didn't score that out specifically, and we were a little confounded early on because some of the gene sets overlap genes that were being expressed by pediatric tumors. So I think to go after that question, we're going to want to go through a similar exercise of, are the exhaustion signatures overlapping with pediatric cancer cell expression profiles? So yeah, it's a great idea. I agree, it probably will be a prognostic signature, and it actually might help us tease apart. I think the piece I'm still confounded by are those survival curves where you have very good PFS in immune infiltrated, but then they don't do very well long term. And I think questions like this might help us understand why that is.
Do you think this could be a prognostic signature for immunotherapy responsiveness?
Yeah, I think it likely will be.
Okay. Brian Crompton asks, should we just add your probes to the tumor-specific panels being developed for paired tumor circulating tumor DNA analysis?
Yeah, thanks, Brian. I mean, I would love to do that. We don't have the data yet, but I suspect that the tumor-reactive T cells are probably enriched in the cell-free DNA. So yeah, it'd be great to team up with you to do that. I think that's where the signal is going to lie, in the cell-free DNA. The other exciting part with cell-free is the fragmentomics analysis, reading out not just cancer cells, but also open chromatin regions associated with immune cells, with T cells and B cells. So there's a lot that can be done with genome-wide cell-free DNA profiling. And yeah, I think baiting out TCR signatures in cell-free DNA would be a pretty interesting experiment.
Another comment come in. Very nice talk. Can you compare the amount of immune infiltration in the pediatric inflamed group to traditionally hot adult tumors like melanoma? Are they inflamed in general or inflamed relative to other pediatric tumors?
Yeah, it's super variable. So there are some tumors that are inflamed to the same level as an adult melanoma and some that aren't. So it's a complete gradient. Pediatric inflamed in our case are in general around the average of adult tumors, but you can tell by those huge spreads on that first colorful immune plot, they stretch all the way up to an ultra hot melanoma-like microenvironment. But in general, pediatric tumors are colder than most adult tumors.
Another question just came in. It says, given that the different sorting methods lead to different results, how do you know which method truly reflects data generated from the protein levels?
Yeah, this is a great question. It's super tricky, especially in genomic test development. What really is the gold standard? Because are we going to benchmark to something that is totally different? We're doing DNA and RNA. It's already well established that correlation between RNA and protein is not necessarily perfect. It held up well in our hands for a select set of proteins. To me, the proof has to be in clinical outcomes. It has to be in measuring a molecule, however you do it, and then showing that that actually has an impact on patient care and patient outcome. So at the end of the day, I don't know if we need to strive for a perfect correlation between all of our biomarkers, but we do need to evaluate all of them in an unbiased and comprehensive way. So we do have that baseline calibration. That's why we set out to do so much of that validation, not necessarily at the single gene or single protein level, but often at the cell type or the gene pathway level.
The questions I had were, what do you think it's going to take for this to become a standard of care?
It's going to have to show some clinical utility. So for that, you're going to have to bake these strategies into clinical trials. And there's going to need to be a lot of infrastructure. So the real challenge is putting genomes and RNA sequencing into practice. Right now, certainly clinical genetics is really in gene panels, but costs are really continuing to fall. And it's actually becoming cost effective to start to be doing genomes and RNA sequencing quite routinely, even on clinically derived specimens. So yeah, I really encourage partnership between clinical lab geneticists and oncologists to set up and scope out those tools, and to encourage something that already happens in pediatric cancers a lot, which is data sharing and multi-site studies. This is the best part of clinical lab geneticists, are these like sample sharing and knowledge sharing across clinical labs who deploy these tests. So I mean, it's happening now. Certainly a lot of our adult trials are really trending quite quickly towards genome and RNA sequencing. And yeah, I think it's going to be infrastructure and partnerships to really make it happen for pediatric cancers as well.
I mean, it seems like it's probably years away. But what do you think?
I think I would say it's single-digit years.
Single-digit years.
This is not decades in the future.
Mm-hmm.
I think we failed if it's going to take that long. But yeah, it's not next month. I think I'd concede that.
Do you think there's even a bigger lift to get this to be a standard of care in like a regional or a community hospital rather than a major urban hospital?
Yeah, this is where relationships between hospitals and reimbursement are really going to come into play, and this is where it's very striking contrast between different jurisdictions. At the end of the day, it's just another molecular test, and that comes with all the current barriers that we already face for targeted gene testing. I don't think even gene testing is all that widely available, so that's a problem to solve totally independently of the genomics platforms. Is how do you open up access, not just to drugs, which is already a challenge, but also open up a challenge to diagnostics. And when you do have a finding, how do you get a kid onto the right drug? So there's actually two access problems to start solving basically now.
Yeah, I mean, so if you run this on a patient and it comes up that they should get a drug that's not available at their hospital, right? So then you have to get them to a hospital that has that drug on trial.
Yeah. And that's where these data networks are so important, because it's knowledge of what have you found and then what trials are actually available. We don't really have Google search for, here's the tumor, what trial could I go on? Like that's actually a pretty interesting piece of infrastructure that really needs to be built.
And then, isn't the insurance company going to want to pay for the family to travel 1,000, 2,000 miles to get the drug, or is it going to be on the shoulders of organizations like Alex's? When we have a travel for care fund that will get families there, but I could see it getting quickly overwhelmed if we have to 10x the number of families that we're helping travel.
Yeah, I mean, I can't speak to what insurance companies will reimburse, but to me, it's money well spent. If you really have an extraordinarily highly predictive biomarker, and this drug's been known to work with very high likelihood, to me, it's going to be money well spent.
I agree. So I don't know if you planned it this way, but our timing has been perfect because we're out of questions and we're out of time. Another question just popped in, so maybe you could take one more. Would this would this need immune infiltrated signature around the tumor and in parentheses biopsy, or would you be able to do the same with peripheral blood sampling?
Yeah, I think that loops back to Brian's question. I think it's in the blood. I have no data to back that up, but from what really, really early work in adults and what's being done with fragmentation data, I think we're going to be able to get it in the blood and to see it over time, which would be really exciting to know, is this original prediction working early, like much earlier? And we do have some data that shows cell-free DNA signal precedes imaging signal. So you could really get quite creative with cell-free DNA plus imaging as both a predictor, but also as a monitor over time as well.
And imagine a brain tumor that you couldn't get a biopsy from, being able to do it for that. That would be incredible.
Yeah, and there's been some great work in CSF as well. So yeah, the biology is there. How you measure it, you can really get quite creative. And yeah, in brain especially, I agree. All
right, well, we're out of time. We're out of questions, but I want to thank you again. It was a great talk, really eye-opening, and I can't wait for the day when we can do this for all the kids that are diagnosed.
Yeah, me too. Thanks a lot, Jay, and thanks everyone for the great questions. Really great discussion.